All right, today we're talking about something huge, literally. We're diving into how scientists figured out how to copy and paste entire chapters from the Book of Life. We're talking about the tech that made things like the Human Genome Project and modern gene therapy even possible. So how in the world do you photocopy a book that's billions of letters long? Let's get into it.
You know, to really get a sense of the challenge here, just think about our own genome. It's a book with about 3 billion letters. Now imagine you need to study one specific chapter of that book, or maybe even a whole volume. How would you even begin to make a copy? That was the exact problem that had geneticists stumped for a long time. So the first cloning tools that came along, things called plasmids, they were totally revolutionary, don't get me wrong, but their carrying capacity, it was tiny. We're talking just a few thousand letters of DNA. Trying to map a whole genome with those was like trying to photocopy an entire encyclopedia using nothing but Post-it notes. It just wasn't going to work. Scientists needed a serious upgrade. They needed a fleet of genetic cargo trucks.
And this brings us right to our first major hurdle, what I like to call the DNA scaling problem. Because in genetics, bigger is very often better. I mean, it's one thing to snip out a single tiny gene, but it's a whole other ballgame to try and understand how entire genetic neighborhoods work together to make us who we are.
So why does this matter so much? Well, because a lot of our genes are absolutely massive, and many genetic diseases are incredibly complex, involving huge stretches of a chromosome. To even dream of tackling something like the Human Genome Project, or to get a full picture of diseases like cystic fibrosis, scientists had to find a way to clone DNA pieces that were hundreds of thousands or even millions of letters long.
The first real breakthrough came from a familiar place, the microbial world. Yep, to build bigger and better DNA carriers, scientists turned to the absolute workhorses of biology, bacteria and the viruses that love to infect them.
The solution was to build something completely new, an artificial chromosome. Basically, it's a synthetic DNA molecule that has all the essential parts of a real chromosome. It's got an origin of replication so it can get copied, a centromere so it gets sorted correctly when the cell divides, and telomeres to protect the ends from falling apart. This design is what allows it to carry these huge DNA inserts and stay stable inside a host cell.
And this idea led to two brand new tools, PACs and BACs. PACs were built using parts from a virus that infects bacteria. BACs came from a special bacterial plasmid. The key thing is that both were designed to be incredibly stable and could carry DNA inserts of up to 300,000 base pairs. That's 300,000 letters of genetic code. It was a massive, massive leap forward.
So how does this P1 system actually work? Well, the vector, our little DNA delivery vehicle, is cut open with incredible precision. This creates a short arm and a long arm, and both of them have what we call sticky ends. These ends are specifically designed to grab onto the huge piece of DNA you want to clone and basically glue it right into place between them.
The whole thing is just a beautiful, clever, four-step process. First, your DNA gets attached to the vector arms. Second, this whole package gets stuffed into the protein shell of a P1 virus. Third, the virus does what it does best. It injects that DNA right into an E. coli bacterium. And then finally, this amazing enzyme called recombinase acts like a molecular lock and key, finds two specific docking sites called LOXP, and just seamlessly stitches the whole thing into a stable circular plasmid.
And right here, you can see that final critical step in action. The virus injects the DNA. Once it's inside, that recombinase enzyme finds those LOXP sites, loops the DNA around, and locks it into a perfectly stable circle. And now it's ready to be copied over and over and over again by the host bacterium. Just brilliant. Now, BACs were the absolute workhorses of the Human Genome Project. But you know how science is? It always pushes for more. What if you need to clone something even bigger? Well, that required moving from bacteria to an even more complex organism. Yeast.
And that brings us to yeast artificial chromosomes, or YACs. And wow, the difference is just staggering. I mean, look at the capacity. A BAC tops out around 300,000 base pairs. A YAC can hold over 1 million. But, and there's always a but, right? There's a trade-off. Because they're so huge and they live inside more complex yeast cells, YACs can sometimes be unstable and actually rearrange the very DNA they're supposed to be carrying.
Okay, so if you're going to build an artificial chromosome from scratch, what do you need? Well, you have to include the 3 non-negotiable parts of a real one. First, you need a centromere, which is kind of like a handle that the cell uses to pull it apart during division. Second, you need telomeres. These are like protective caps on the ends to stop them from fraying. And third, you need origins of replication, the start copy here signal.
The strategy they came up with was just ingenious. They started with a circular plasmid in bacteria that already contained all those essential chromosome parts. Then they used enzymes to cut it open, which created a left arm and a right arm. And that massive piece of DNA you want to clone, it gets slotted right into the middle, creating one single enormous linear artificial chromosome.
But wait, how do you find the few cells that actually took up the new DNA? Well, the scientists built in this incredibly clever visual trick using a special marker gene. The spot where the new DNA gets inserted is right in the middle of this gene. So if a big piece of DNA successfully gets in, it breaks the gene, and the yeast colony grows white. If the vector just seals back up on itself without the insert, the gene stays intact and the colony turns red. It's a simple, brilliant color code for success. Okay, so we've figured out how to clone huge chunks of DNA in bacteria and in yeast. But what about getting genes into animal cells, including our own? For that job, scientists turned to nature's ultimate delivery experts, viruses. So instead of just fighting viruses, researchers had this incredible realization: what if we could repurpose them? Viruses have spent millions of years perfecting the art of getting their own genetic material inside of cells. So, scientists basically figured out how to hollow them out, remove all the harmful bits, and turn them into these high-tech molecular delivery vehicles or vectors.
And there is a whole fleet of them to choose from. This table just shows a few of the key players. Now notice their insert size is way smaller than BACs or YACs. These are for delivering specific genes, not entire chapters. But the other really key difference is whether or not they integrate into the host genome. Retroviruses will actually stitch their DNA right into your chromosomes for a permanent change, while others, like adenovirus, just deliver their cargo as a temporary, separate piece of DNA.
Now this diagram shows a really crucial safety feature. Because you might be thinking, isn't using a virus a little dangerous? Well, to prevent the engineered virus from just replicating like crazy, scientists use a special packaging cell. This cell is given all the instructions to build the viral parts, the shell, the enzymes, but those instructions are not included in the final virus itself. The only genetic material that gets packaged is the good stuff, the therapeutic gene. The result is like a high-tech delivery drone. It can infect a target cell one time, drop off its cargo, but it can't make copies of itself. It's a one-way trip, which is exactly what you want.
So, after this whole journey from Post-it notes all the way to viral delivery systems, you can really start to see how scientists have put together this incredibly powerful and diverse set of tools.
This final table really puts it all together. This is the geneticist toolkit. If you need to map out a genome with super high fidelity, you grab a BAC. If you're studying some monstrously large gene, you reach for a YAC. If you need to permanently add a gene to a human cell for gene therapy, a retrovirus is your tool. See, there's no single best vector. There's only the right vector for the job you need to do. And that's really the core takeaway here. Modern genetics isn't about finding one single magic bullet. It's about having this sophisticated and specialized toolkit where every single instrument, from a bacterial chromosome to a repurposed virus, is perfectly suited for a very specific scientific task.
This technology has given us an absolutely unprecedented ability to not just read, but to write and edit the code of life. And that leaves us with a final, massive question. Now that we have these incredibly powerful tools, what new cures, what new discoveries, and what new biological stories will we choose to write next?